In this video, we'll discuss the molecular mechanisms of insulin resistance due to obesity. The insulin receptor is a dimer made of two extracellular alpha subunits and two transmembrane beta subunits. Disulfide bonds connect the alpha and the beta subunits, as well as the two alpha subunits, to maintain the receptor as a dimer. Insulin binds into this cavity created by these two alpha subunits of the insulin receptor, which face the extracellular environment. Now the alpha subunits move closer together and lock the insulin between them. This also causes the beta subunits to move closer. This results in a conformational change of the protein, which activates the tyrosine kinase domains on each beta subunit. A tyrosine kinase is an enzyme that phosphorylates tyrosine amino acids on target proteins and enzymes. The tyrosine kinase activity causes the autophosphorylation of several tyrosine residues on the opposite tyrosine kinase domain. The process of phosphorylating each other is called cross-phosphorylation. Both these beta subunits are phosphorylated in the presence of ATP. The phosphorylation of three residues of tyrosine is necessary for the amplification of the kinase activity. The tyrosine kinase domains can also phosphorylate other proteins. One of the phosphorylated tyrosine residues on the insulin receptor, specifically this one over here, attracts a protein called the insulin receptor substrate 1, or simply IRS1. IRS1 gets phosphorylated by the tyrosine kinase domains. The phosphorylated sites on IRS1 serve as attachment points for a lipid kinase called phosphoinositide 3 kinase, or PI3K for short. Lipid kinases are kinases that phosphorylate some type of lipid molecule. In our case, this lipid molecule is called phosphatidylinositol 4,5-bisphosphate, or PIP2. PIP2 is located in the cell membrane. You can see it right here. PI3K phosphorylates PIP2. The addition of phosphate converts PIP2 to PIP3. Here I'll show the full chemical reaction of the conversion of PIP2 to PIP3. PIP3 stands for phosphatidylinositol 3,4,5-triphosphate. PIP2 gets phosphorylated by the PI3K kinase. Number 2 in PIP2 means there are two phosphates in the molecule. Number 3 in PIP3 means there are three phosphates in the molecule. The additional phosphate will be added right here at the third carbon. Now back to the cell membrane. The newly formed PIP3 can move along the membrane and serve as a recognition site for an enzyme called 3-phosphoinositide dependent kinase 1, or PDPK1 for short. PDPK1 phosphorylates the inactive form of protein kinase B, also known as AKT, and now you can see the chemical reaction. The phosphorylation activates protein kinase B. Here you can see a vesicle with glucose transporters embedded in its membrane. These glucose transporters are called GLUT4, which stands for glucose transporter type 4. Activated protein kinase B, or AKT, phosphorylates a protein called AS160, which stands for AKT substrate of 160 kilodaltons. Here you can see the chemical reaction. This phosphorylation inhibits the GTPase activating domain associated with AS160. Now let's take a look at the AS160 before the reaction, when the GTPase activating domain was still active. In the activated state, the GTPase activating domain is able to activate an enzyme called GTPase. The GTPase binds to GTP and hydrolyzes it to GDP. Now let's take a look at the phosphorylated AS160 after the reaction. The GTPase activating domain is now inhibited, so there's no one around to activate the GTPase, which is located in the same place. If there's no one to activate it, that means the GTPase is also inhibited. As a consequence, GTP is not hydrolyzed to GDP. I didn't mention that this GTPase is part of a protein called RAB. Rab is the next protein in this cascade. You can see it in pink. I've already explained why GTP is not hydrolyzed to GDP. So to summarize the big picture of this process, Rab changes from its GDP-bound state to a GTP-bound state, which activates it. Activated Rab gets attached to this vesicle and will carry it to the plasma membrane. This vesicle will fuse with the cell membrane. GLUT4 transporters will translocate to the cell membrane. Right now, we're in the fed state. That means our blood glucose levels are high, as I previously told you. I will denote glucose with this capital G. These transporters will perform the uptake of glucose from the blood into the cytoplasm. It's imperative to understand the normal insulin transduction pathway in order to understand the mechanisms of insulin resistance induced by obesity. Obesity is accompanied by increased plasma concentration of free fatty acids, leading to their intensified uptake by tissues involved in the regulation of glucose homeostasis, such as the skeletal muscles, the liver, and the pancreas. In order to see the distribution of the adipose tissue, let's take a look at the sagittal section of the abdomen and the thigh in an obese patient. The adipose tissue beneath the skin is called subcutaneous adipose tissue, or SAT for short. The adipose tissue lining the internal organs is called visceral adipose tissue, or VAT for short. The source of free fatty acids in the blood can be both the subcutaneous adipose tissue and the visceral adipose tissue. Fatty acids form through a process called lipolysis. Lipolysis is the metabolic process through which triacylglycerols break down via hydrolysis into their constituent molecules, which are glycerol and free fatty acids. Due to the higher lipolytic activity of visceral adipocytes, the release of free fatty acids into the blood from visceral adipose tissue is faster and in greater amounts compared to the subcutaneous adipose tissue. This was observed in both obese and lean patients. At the beginning of the video, I told you that obesity is accompanied by increased plasma concentrations of free fatty acids, and now you know why. If you have a lot of adipose tissue, you'll have a lot of lipolysis, and a lot of free fatty acids released into your blood. It's simple. I also told you that increased plasma concentrations of free fatty acids leads to their intensified uptake by tissues involved in the regulation of glucose homeostasis, such as the skeletal muscles, the liver, and the pancreas. Now let's go back to the insulin transduction pathway. You can see that the concentration of free fatty acids is increased. Our cells have a way to deal with excessive amounts of free fatty acids inside them. Beta oxidation is the catabolic process by which fatty acid molecules are broken down in the mitochondria to generate acetyl-CoA. Fatty acid beta oxidation is the process by which fatty acids are broken down to produce energy. Increased fatty acid uptake, exceeding the cell's ability to oxidize them in the mitochondria, leads to the intracellular free fatty acid accumulation. All these excessive amounts of free fatty acids floating around will lead to the development of a phenomenon called lipotoxicity. Lipotoxicity favors the reduction of insulin sensitivity and gradual atrophy of the beta cells in the pancreas, and we'll see the mechanism in just a second. Lipotoxicity is a metabolic syndrome that results from the accumulation of lipid intermediates in non-adipose tissue, leading to cellular dysfunction and potentially cell death. The tissues normally affected by lipotoxicity include the kidneys, the liver, the heart, the skeletal muscles, and the beta cells of the pancreas. Lipotoxicity is believed to have a role in heart failure, obesity, and diabetes, and is estimated to affect approximately 25% of the adult American population. In the past, it was assumed that insulin resistance was induced by triacylglycerols that accumulate inside the cells. However, attention is currently focused mainly on biologically active lipids, capable of inhibiting or activating enzymes that directly affect the insulin pathway. These lipids include long-chain fatty acyl-CoA, ceramides, and diacylglycerols. Firstly, let's focus on fatty acids and long-chain fatty acyl-CoA. After passing through the cell membrane, fatty acids are activated by attaching coenzyme A in a reaction catalyzed by an enzyme called long-chain fatty acyl-CoA synthetase in the presence of ATP, resulting in the formation of long-chain fatty acyl-CoA, AMP, and pyrophosphate. Literature data indicate that long-chain fatty acyl-CoA levels in obese people increase not only in the tissues affected by lipotoxicity, but also in the adipose tissue. Activated fatty acids in the form of long-chain fatty acyl-CoA are substrates in the de novo synthesis of other lipids, such as triacylglycerols, diacylglycerols, and ceramides, or they can undergo beta oxidation in the mitochondria. Long-chain fatty acyl-CoA, diacylglycerols, and ceramides are considered biologically active lipids, or bioactive lipids. There are a lot of bioactive lipids. Bioactive lipids in general play an important role in energy homeostasis, cell proliferation, metabolism, inflammatory homeostasis, and other functions. However, these three bioactive lipids mentioned above play an important role in the development of insulin resistance. Let's start with the long-chain fatty acyl-CoA and how they generate insulin resistance. It has been demonstrated that long-chain fatty acyl-CoA activates some isoforms of protein kinase C, or PKC for short. As a side note, a protein kinase is a kinase that selectively phosphorylates other proteins. Here you can see how a protein kinase phosphorylates a certain protein in the presence of ATP. Protein kinase C is involved in controlling the functions of other proteins through the phosphorylation of hydroxyl groups of serine and threonine amino acid residues on these proteins. Here you can see the chemical reaction. The hydroxyl groups that get phosphorylated are in yellow. Now we're ready to discuss the molecular mechanisms of insulin resistance due to high levels of long-chain fatty acyl-CoA. As I've already told you, it has been demonstrated that long-chain fatty acyl-CoA activates some isoforms of protein kinase C. Protein kinase C will come near this molecule, called the insulin receptor substrate 1, or IRS1 for short. Now let's zoom in on this molecule. Each green circle represents an amino acid from the IRS1 molecule. Let's add some numbers to these amino acids, which will signify their order. Now you can see the first 12 amino acids. The 3 dots represent some omitted amino acids in this long chain. After the 3 dots, you can observe the 307th amino acid residue in this protein, which is serine. Protein kinase C will phosphorylate this serine residue. Now you can see the phosphate group. I added numbers so you can see the amino acids from 307th to 316th. IRS1 is a 131 kilodalton protein with an amino acid sequence of 1242 residues. So the last amino acid in this chain will be numbered 1242. I'll also add numbers to the amino acids that precede it. These numbers are totally random, so don't take them into consideration for what I'm about to show you. Normally, multiple tyrosine residues of IRS1 are phosphorylated by the tyrosine kinase domains of the insulin receptor. This is a part of the insulin signaling pathway, and you can see the same phosphate groups on this side of the picture. The phosphorylation of IRS1 at serine 307 by the protein kinase C blocks the phosphorylation of tyrosine residues on IRS1, which is performed by the tyrosine kinase domains of the insulin receptor. That's not good, and you'll see why. Let's take a look at the big picture. Initially, IRS1 had 4 phosphate groups, but now it has only 1. If IRS1 is not properly phosphorylated by the tyrosine kinase domains, the binding and activation of phosphoinositide 3 kinase necessary for the continuation of the insulin transduction pathway will be impossible. As a consequence, the whole insulin transduction pathway will not be properly activated. This is a huge problem, because all the functions of insulin inside our cells will suffer. For example, insulin won't be able to increase the glucose uptake in the target cells. As you can recall from the previous video, protein kinase B has other functions besides increasing glucose uptake. You can see how protein kinase B phosphorylates specific enzymes. These enzymes can phosphorylate or dephosphorylate specific metabolic enzymes, which can stimulate or inhibit different metabolic pathways inside our cells. As you can recall from the previous video, the main functions of insulin inside the cell are to stimulate glycogenesis and inhibit glycogenolysis. It stimulates glycolysis and decreases gluconeogenesis. Insulin also stimulates the synthesis of fatty acids from non-lipid precursors, stimulates lipogenesis, and decreases lipolysis. In the muscles, insulin can also increase the expression of numerous amino acid transporters. That's how insulin can increase amino acid uptake in the muscle cells. Now that we have a lot of amino acids inside the cell, these metabolic enzymes will stimulate the synthesis of proteins. These enzymes will also decrease proteolysis. Insulin can also decrease autophagy. All the functions mentioned above will suffer. When your muscle cells, adipocytes, hepatocytes, and other cells in your body don't respond properly to insulin, resulting in elevated levels of glucose in the blood, you have insulin resistance. As I previously told you, long-chain fatty acyl-CoA, diacylglycerols, and ceramides are considered biologically active lipids, or bioactive lipids. Now let's move on to the next chemical compounds that can affect insulin resistance, called diacylglycerols. Diacylglycerols may be formed in a cell as a result of the breakdown of phospholipids or in the de novo synthesis. Let's start with the de novo synthesis of diacylglycerols. We'll start with glycolysis. Glycolysis is the process in which glucose is broken down within the cytoplasm of a cell to form pyruvate. Glycolysis has 10 steps, so it has a lot of intermediates. It produces 2 molecules of pyruvate, ATP, and ADH. Diacylglycerol synthesis starts with a very important intermediate from glycolysis, which is dihydroxyacetone phosphate, or DHAP for short. Dihydroxyacetone phosphate gets reduced in the presence of NADH to form glycerol 3-phosphate. The reduction is catalyzed by glycerol 3-phosphate dehydrogenase, or GPDH for short. The key stage of the de novo synthesis of diacylglycerols is the glycerol 3-phosphate acylation, catalyzed by glycerol 3-phosphate acyl transferase, or GP81 for short. The reactants of this reaction are 1 molecule of glycerol 3-phosphate and 1 molecule of long-chain fatty acyl-CoA. The direct product of this reaction is lysophosphatidic acid. Studies have shown that overexpression of GP81 can lead to steatosis and increased insulin resistance, and you'll understand why in just a second. If we add another long-chain fatty acyl-CoA to the lysophosphatidic acid, it gets converted to phosphatidic acid. Now let's remove the phosphate group from the phosphatidic acid and turn it into diacylglycerol, or DAG for short. Okay, we're done with the de novo synthesis of diacylglycerols. Diacylglycerols may also be formed in a cell as a result of the breakdown of phospholipids by an enzyme called phospholipase C, or PLC for short. Here you can see a generic phospholipid. Phospholipase C is a class of membrane-associated enzymes that cleave phospholipids just before the phosphate group. Right here. A new molecule of diacylglycerol is synthesized. In the cell membrane, we have a phospholipid called phosphatidylinositol 4,5-bisphosphate, or PIP2 for short. You can see its chemical formula. The hydrolysis of PIP2 occurs as a result of the action of phospholipase C, which will cleave this phospholipid just before the phosphate group. Right here. Phospholipase C will cleave PIP2 into diacylglycerol and inositol 1,4,5-triphosphate, or IP3 for short, which serve as second messengers. So, this is the second method to synthesize diacylglycerols. Now, we're ready to discuss the molecular mechanisms of insulin resistance due to high levels of diacylglycerols. Excessive accumulation of diacylglycerols leads to the activation of certain isoforms of protein kinase C that are responsible for abnormal insulin signaling. The mechanism of insulin resistance due to high levels of diacylglycerols is the same as that induced by high levels of long-chain fatty acyl-CoA. So, diacylglycerols will activate protein kinase C. The phosphorylation of IRS1 at serine 307 by the protein kinase C blocks the phosphorylation of tyrosine residues on IRS1, which is performed by the tyrosine kinase domains of the insulin receptor. If IRS1 is not properly phosphorylated by the tyrosine kinase domains, the binding and activation of phosphoinositide 3 kinase necessary for proper insulin action will be impossible. As a consequence, the whole insulin transduction pathway will not be properly activated. This is a huge problem because all the functions of insulin inside our cells will suffer. For example, insulin won't be able to increase the glucose uptake in the target cells. When your cells don't respond properly to insulin, resulting in elevated levels of glucose in the blood, you have insulin resistance. In vitro studies on isolated skeletal muscle cells and adipocytes have shown that the activation of specific isoforms of protein kinase C by diacylglycerols leads to an impaired GLUT4 translocation to the cell membrane and reduces insulin-dependent glucose uptake. Intramuscular accumulation of diacylglycerols was found in insulin-resistant rats fed with high-fat diet. Moreover, a threefold increase in liver content of diacylglycerols was observed in rats infused with a lipid emulsion called intralipid, resulting in the inhibition of the insulin pathway. Lipid emulsions, or fat emulsions, refers to an emulsion of lipids for human intravenous use, especially for feeding critically ill patients that can't consume food and administer their required nutrients entirely. As a side note, intralipid is a lipid emulsion with the following composition: soybean oil, egg phospholipids, and glycerin. This emulsion is available in 10%, 20%, and 30% concentrations. In the next video, we'll discuss other chemical compounds that can affect insulin resistance, called sphingolipids and ceramides. Good luck!